Welcome to Biology at Priyojit, my dear viewers. Today we are going to explore regulation of transcription in eukaryotes and understand how eukaryotic cells precisely control gene expression. We learn how activators and enhancer molecules switch genes on and how repressors and silencers turn genes off, and how chromatin structure and DNA methylation influence the process of transcription. Then we will move beyond transcription to discover how siRNA and miRNA mediate gene silencing at the post-transcriptional level. By the end of this lecture, you will clearly understand transcriptional regulation and RNA interference at a simple UG-PG conceptual level. So let's break down this fascinating regulatory system step by step. So, what is gene regulation? Gene regulation is basically the control of gene expression so that genes are expressed at high, low or no levels depending on the need of the cell. So, why the process is important? Eukaryotic organisms like protists, fungi, plants, animals regulate genes to adapt to environmental changes. It responds to nutrient availability. It protect against environmental stress like UV radiation, and it control development and differentiation. So as the example, the tanning response in human protects skin cells from UV damage. So this is the image showing the different levels of gene expression commonly subject to regulation in eukaryotes. There is a transcriptional control, RNA processing, there is regulation in translation and post-translational modifications. So levels of gene regulation eukaryotes like prokaryotes, eukaryotes can regulate gene expression at multiple stages. We have already mentioned in the last image, transcription, RNA processing, RNA transport, mRNA stability, translation and post-translational modification. These are the steps where gene expression can be regulated. So basically most regulations occurs at transcription initiation. Transcription factors, they are the protein factors that influence RNA polymerase activity. They basically regulate binding of RNA polymerase to promoter and transition from initiation to elongation. Types of transcription factors, there are basically general transcription factors which are required for RNA polymerase to bind core promoter and necessary for basal level transcription, present in all actively transcribed genes. So without general transcription factors there is no transcription. Prokaryotic, there is regulatory transcription factors, increase or decrease of transcription rate, bind to specific DNA regulatory elements and responsible for differential gene expression. Here the general transcription factors or transcription factors binds the promoter, and RNA polymerase also bind at the promoter region, and then transcription begins. So regulatory element which act as cease acting elements is the DNA sequence that regulate the transcription. Enhancer increases transcription and silencer element decreases transcription. These elements are usually near the promoter, but it may be upstream, downstream or inside the gene. Thousands of base pairs are away. Activators and enhancers. Activator is the protein that increases transcription rate. This is the image showing the transcription process. Enhancer is the DNA sequence where activator binds. So how it works? Activator binds enhancer, DNA then bends, and activator interacts with promoter bound proteins. Then RNA polymerase is recruited. Then transcription is increases. So enhancers are often orientation independent or bidirectional process. Example include forward 5' GA TA 3' and the reverse sequence is the 5' TA TC 3'. So both the sequence enhance transcription. Repressor and silencer. The protein repressor is the protein that can inhibit transcription, that can downregulate the transcription process, and silencer is the DNA sequence where the repressor protein can bind. So what is the mechanism? Repressor blocks activator binding and it prevents RNA polymerase binding with the promoter sequence. It recruits chromatin modifying enzymes. So what are co-repressors? So this is the image showing the enhancer. sequence and activator protein which increase the rate of transcription, and here is the silencer sequence and repressor protein which downregulate the transcription process. So what is co-repressor? Co-repressor is also protein that cannot bind DNA directly, but they bind transcription factor and represses the gene expression. Mechanism: it recruits histone deacetylase or HDAC, which removes acetyl group from the histone protein, and it increases positive charge of the histone protein so that the DNA can bind tightly to the histone and chromatin becomes compact and transcription decreases. So what do you mean by combinatorial control? Most eukaryotic genes are controlled by multiple factors simultaneously. This is called combinatorial control. Factors involved multiple activators, multiple repressors, effector molecules, protein-protein interactions, covalent modifications, chromatin remodeling, DNA methylation, and this expression depends on the combination of all signals. So this is the activator and the molecules, then it is repressor. So this gene is only expressed if both activators are present and the repressor is absent. So when both activators are present but repressor are absent, then transcription is going on. But when activators are present but repressor is absent, only activator is present, so then little or no transcription. Only one activator is present. But when both activator and repressors are present, activator is present but repressor is still present, so there is no transcription. So this is the combinatorial control of gene expression. DNA methylation: addition of the methyl group to the cytosine nitrogen base or nucleotide CpG island. It often causes gene silencing. It prevents activator binding, or it recruits protein that compact the chromatin, and it results reduced rate of transcription. Structure of transcription factors: the transcription factors control functional regions called domains, and these common domains have DNA binding domain and activation repression domain and ligand binding domain. Motif: it is a structural pattern repeated in many proteins. Example: zinc fiber, helix turn helix, and leucine zipper. Here is the activator, it binding site for this activator, and activator helps generate transcription factor and RNA polymerase assembly. So methylation of DNA and histone causes nucleosomes to pack tightly together, and transcription factors cannot bind the DNA and genes are not expressed. siRNA mediated gene silencing. siRNA is the small interfering RNA that is approximately 21 to 23 nucleotides. So it is similar to miRNA but different origin and function. siRNA mechanism: double-stranded RNA processed by the protein dicer, then siRNA is loaded into the RISC complex, protein complex, then one strand is retained or guides, which is guide strand, and both and binds perfectly to the complementary mRNA. Then AGO2 protein cleaves the mRNA and mRNA is degraded, so it results stronger gene silencing. So what do you mean by miRNA mediated gene silencing? miRNAs are small non-coding RNAs that is approximately which has 22 nucleotides. So its function is post-transcriptional gene regulation. miRNA mechanism: miRNA associates with argonaut or AGO protein. It forms miRSC, miRNA induced silencing complex. It binds target mRNA. So this is the image showing the siRNA mediated gene silencing, and this is this image showing how the mRNA binds with the partially complementary sequence of the mRNA and immediate gene silencing process. So if perfect complementary, then mRNA cleavage occurs, which is rare in animals, but if partial complementary, so which is common in animals, so it translational repression, deadenylation, decapping, and 5' to 3' degradation occurs. So important points include AGO proteins: only AGO2 is catalytically active in humans, GW182 proteins and deadenylase complexes PAN2, PAN3 and CCR4 not. So importance of miRNAs: it regulates more than 50% of human protein coding genes. They are involved in cell growth, cell differentiation, cell development and metabolism, and dysregulation of miRNAs causes cancer and disease. So we can conclude that regulation of transcription in eukaryotes is highly coordinated and multi-layered process. Activators and enhancers work together to increase transcription. while repressor and silencer reduce or completely block gene expression. Chromatin modifications and DNA methylation further influence whether a gene remains active or silent. Beyond transcription, siRNA and miRNA provide powerful post-transcriptional control by degrading mRNA or inhibiting translation. Together, these mechanisms ensure precise, flexible, and efficient gene regulation, which is essential for development, differentiation, and cellular homeostasis. So these integrated control system makes eukaryotic gene regulation a fundamental and fascinating topic for clear undergraduate and postgraduate level understanding. Thank you for watching this video.